Alright, we're going to do a quick overview of how bacteria become resistant to antibiotics. The focus is going to be on Neisseria gonorrhea due to the case this week. However, I want to say really quick that I read a few review articles in Nature and New England Journal of Medical Medicine, and what I saw over and over again was that it's really important to point out that it's important to know that things become resistant. So this is me doing diligence. Hey, it's really important to know about why bacteria become resistant because it can cost a lot of money, people will perish, and quality of life will quickly lessen. Okay, the paradigms. If you have a microbial infection, the first thing you do is you're going to get your antibiotics. You're going to take your antibiotics and you have to get the antibiotic into the cell. So the antibiotic is going to cross through the cell membrane. It's going to get inside the cell, and then when it's inside the cell, it's got to build up its concentration. When the concentration is high enough, then it can attack its target. And hopefully as a result of that, what we're going to see is that the cell will die. So resistance is simple. It's basically just the bacteria's strategy to prevent the drug from doing what it does. And so it can do that in a couple of ways. You can reduce the intracellular concentration of the drug. You can do that from blocking the drug from getting in, or once it gets in, you can pump it back out. The other thing is you can alter its target protein. And so however you do that, you can alter it by either having a mutation to the protein itself, the target itself, or you can post-translationally modify it. So post-translational modification would be like you block the drug from having access to it. And there's more than one way to do that, so you could basically move the target to a place where the drug doesn't have access. You can inactivate the drug itself. And then two other things that aren't always mentioned in every book that you need to be aware of is that you can have an alternate metabolic pathway, or you can have phenotypic variation. And I'm going to explain what both of those things mean. First, really quick, how do you get antibiotic resistance? So back in the 1930s, most Staphylococcus did not have resistance to penicillin. Now today, most Staphylococcus aren't even affected by penicillin. So there's a couple ways. You can have intrinsic resistance. Intrinsic resistance is like saying, what is the spectrum of antibiotic activity? Vancomycin, for example, has no activity against gram-negatives, so gram-negative bacteria are intrinsically resistant to vancomycin. The raw material for all resistance is basically spontaneous mutation. Eventually, spontaneous mutations accumulate in one bacteria, and then it starts sharing that stuff with everybody else, and that is the raw material for everything. Because once you've got that spontaneous mutation, then you can start transferring the genes, called horizontal gene transfer, where you have this bacteria here and this bacteria here, and this one decides that he's going to share some of his chromosomal information. That's horizontal transfer. This is not his kid, it's his buddy, and he's just like, hey, I'm going to share my DNA with you. You already know from your micro that it happens via conjugation, transformation, and transduction. But other than changes to DNA, you can also have phenotypic variation. And phenotypic variation would be things like Bacillus anthracis. If you give it time to form a spore, that spore is going to be resistant to almost any antibiotic. It is resistant to any antibiotic that you can put at it. Another thing is called quorum sensing. There are some bacteria that they will just turn off their genes for a cell wall. They're gram-positive bacteria that they will just say, I don't want my cell wall anymore, and in certain situations they can do that. Quorum sensing also is where you have a whole group, a whole community of bacteria living together, and they're signaling to each other, and the signaling tells them, hey, turn off these genes, turn on those genes, and by changing the gene expression, they're no longer sensitive to some of the antibiotics. So intrinsic resistance. It used to be thought that Pseudomonas was resistant to triclosan because of an efflux pump. It was recently discovered, however, that they don't have an efflux pump for triclosan. They just don't have the target that triclosan attacks. Gram-negative bacteria are resistant to daptomycin. You'll remember that daptomycin, it likes to take insert basically a tail into the cell membrane. This basically disrupts the cell membrane, causes it to lyse. The mechanism that it uses is that it has to take a calcium-mediated activity. It inserts into an anionic phospholipids. Gram-negatives have less anionic phospholipids, and therefore they are resistant. Okay, last of all, gram-negative resistant to glycopeptide antibiotics. I'm going to try to draw a gram-negative bacteria so that I can explain what I'm talking about. So here is my gram-negative bacteria. And then you'll remember on the outside of the gram-negative bacteria, so if I say that this is the gram-negative cell membrane, you'll remember that it will have another cell membrane right here, and then in the midst of that it will also have some cell wall, just not very much cell wall, so it'll have some cell wall here as well, and this is called the periplasmic space. So the way that antibiotics get into the gram-negative bacteria, there are things called porins, and those porins will allow the gram-negative bacteria to move nutrients in, and the antibiotics will just move in along for the ride. But glycopeptide antibiotics, like vancomycin for example, they're huge. They're way too big to fit through the porins, and so the gram-negative bacteria are intrinsically resistant. And like I said, when you're talking about intrinsic resistance, you're basically saying what is the spectrum of activity of this antibiotic. And so I pulled this table off the internet. I sent a few emails trying to get permission to use it. I haven't gotten any reply back, so hopefully this isn't an issue. But you look at the spectrum of activity. For example, the spectrum of activity of ertapenem. It covers everything except for the Pseudomonas. Meropenem, imipenem is supposed to cover everything. Now we know it doesn't actually do that. There are things called carbapenemases that will destroy and prevent the imipenem, meropenem, other carbapenems from attacking them. But take for example, if you know you have a gram-positive infection, you're going to be using something that's highlighted right here. On the other hand, if you have a gram-negative infection, you know you're going to be using one of these drugs. And we always say that if you have an anaerobe above the diaphragm, clindamycin, below the diaphragm, metronidazole. Okay, so let's elaborate a little bit on the permeability. So if we can reduce the intracellular concentration of the drug that's getting inside of the bacteria, then the bacteria won't be harmed. Now if I got a bacteria right here, and let's suppose that maybe one molecule of antibiotic gets into it. The bacteria is still not going to die. It's not going to be inhibited. 2 molecules get into it. It's still going to be okay. 3 molecules get into it. Eventually you're going to have a problem. As long as you understand that a few molecules of antibiotic can get inside the bacteria and it will still be okay, but there is some kind of critical threshold. There's like an ED50. Now when we're talking about porins and when we're talking about efflux pumps, most of the time we're talking about gram-negative bacteria. When you're talking about porins, 99% of the time, you're talking about gram-negative bacteria. When you're talking about efflux pumps, about 70 to 80% of the time, you're talking about gram-negative bacteria. Now you can down-regulate the number of porins that you express. This can be done through a number of things. You can have a mutation in the promoter region, or you can just have phenotypic variation. You can also have mutation in the porin protein itself, which will not allow a certain threshold of size of a molecule to get in. Now let's talk about efflux pumps. That's another way we can keep the threshold down. And so the efflux pumps, what you got to know is that there are narrow spectrum efflux pumps and there are broad spectrum efflux pumps. The broad spectrum are called multi-drug resistant. And what you also need to know is that there are several classes of multi-drug resistant efflux pumps. There's not just one, there are several different classes, and each class has subclasses even. The best studied efflux pump is the RND class. The RND class includes the subclass, the ACR from E. coli and the MEX from Pseudomonas aeruginosa. Okay, and the rest of this we're going to cover in the picture I've drawn. Okay, so here's my efflux pump, and what you need to know about it is that there are 3 major components of the efflux pump. There is the basilar or the bottom component, there is the adapter component, and then there is the channel component. And we're actually just lucky that the most popularly studied efflux pump is set up to have this named as a B, this one as an A, and this one as a C, because they're not all named like that. I'm just going to go ahead and tell you. But the easy way to remember it is to remember that it's A, B, and C, or C, A, B, however you want to remember it, but just remembering these. Okay, so another thing I want to point out is that this basilar part, this is a homotrimer. I couldn't draw in 3-dimension, but basically there's supposed to be 3 pieces to it, and they're all supposed to look exactly the same. And what you need to know about this is that there is this distal portion that has a certain size, and then there is this upper portion that has a different size. And so what I'm trying to say is that this efflux pump can carry multiple sizes of things because of the way that this basilar system is set up. Now I'll go ahead and admit that this isn't drawn perfectly because on analysis, like I said, that this is a homotrimer. There are 3 of these, 6 of these proteins, and 3 of these. It's a ratio of 3 to 6 to 3. Okay, I'm going to get a little bit deep, but I'm just going to get you oriented in case you ever go and start reading any type of review papers. This was a little bit of trudging before I finally caught a hold of everything. So there are multiple kinds of adapter proteins. For example, the E. coli would have one called the AcrAB type A. So it has this A, this prefix refers to the A component, or the MEX type A, that would be with Pseudomonas. And then down here you have this AcrB, the MEXB. And then up here at the top you have the TolC, the C referring back here, and this OPRM. This is a completely different bacteria. I'm just giving examples at this point of outer membrane channels. Now another thing I said is that this has different sizes, and so it can carry a lot of different things, but you have to realize that every single enzyme in the world, or every single carrier in the world, has a specific Km, or you might call it a dissociation constant, for the particles or the things that it's going to carry. And so you can actually do minor alterations in these carrying proteins to give it more specificity for one antibiotic over another one. Okay, and I pointed out what all of these things are, but what you need to know is when it's all put together, when this whole thing is put together, suppose we're looking at an E. coli bacteria, the whole thing will be called the AcrAB-TolC. This is a subtype of the RND. The RND is a subtype of the multi-drug resistant transporter. I'll show you a big picture here in a second. So we're coming back out of the woods now. I've already pointed out that there are 2 different sizes here. And I've said that mutations in these sites can change the specificity for one antibiotic over another. Now here's the really interesting thing about multi-drug resistant proteins is that almost every kind of bacteria, at least almost every kind of gram-negative bacteria, has these in their genome already. There's actually been some identified in Staphylococcus and other gram-positives as well. But almost every kind of gram-negative already has these. They just aren't using them to pump drugs out. The reason for that is because they're controlled at the genetic level. So you'll see that at this place right here, I have this promoter. The promoter for this is called the MTR capital A. The MTRA promoter is something that Neisseria gonorrhea has. I'm going to start talking about Neisseria gonorrhea because this is where it starts coming out of the woodwork. So if I were to transcribe this gene right here, what I would make is a protein. That protein would be a transcription factor called MTRA. On the other hand, if I were to transcribe this gene, it would produce a protein. That protein would be a repressor for the exact same gene that this is a transcription factor for. And yes, these genes on the bacterial genome are side by side. Now at this point I'm talking about there are regulators. So we're talking about regulators. You got to know that the regulators can be global, they can also be local. So for example, this MTR regulator, it might control several genes, and one of those would be part of the multi-drug effluxes. Now that's too deep in the woods to really worry about. You may come across in your reading something that says ARAC-XYLS family, the ARAC-XYLS. That's just the family of these transcription factors. So if the balance is favoring my repressor, then of course I'm not going to make the genes for the multi-drug resistant. But if my balance is favoring the transcription factor, then eventually I'm going to go over down further in my DNA, bind to that, and create the proteins for the multi-drug resistance. And so whenever you're talking about the Neisseria gonorrhea, it's the MTRA promoter, because you're going to make MTRC, MTRD, and MTRE. Those are going to be the genes that do this. So it will no longer be CAB, but this is just one of those weird exceptions. You should also know that each one of these proteins, I've given you the names that I've seen in review articles and stuff, but they also have other names. If you do a quick internet search for any of these, you're going to not find necessarily what you're looking for, because they have 2 or 3 names sometimes. And so here's a really sky view look at the whole thing as far as efflux. So you have several different families. I told you the RND is the most studied one. It is a multi-drug efflux pump. There is another one, LMRA, that's a multi-drug efflux pump. And then these over here are specific for specific families of antibiotics. So I told you there are repressors and there are transcription factors, and we know that sometimes the repressors are winning and sometimes the transcription factors are winning. And what causes that to happen? For the most part, we could say mutation. So mutation caused that to happen. So in Neisseria gonorrhea, there's a mutation at the -10 consensus sequence for the MTRC protein, and whenever that mutation happens, it overexpresses and you get overproduction of the multi-drug resistant. So mutation, and the other one is environmental factors. So for example, environmental factor, E. coli and Salmonella will upregulate their MDR transcription factors in the presence of bile, and also in the presence of indole. Their MTR, CDE, and Neisseria, and also the NORA and Staph aureus respond to iron limitations. So if you have a lot of hepcidin, you're storing away all your iron, and that lack of iron causes them to start expressing the multi-drug resistance gene. Now at this point you're probably thinking Neisseria is probably like a badass or something. You're really right, but not for the reason that it has this really cool efflux pump, because there are studies that show, here it is, the study that shows that Neisseria would not even survive in your body. You could take no antibiotics, you could take nothing. Neisseria still wouldn't survive in your body without its multi-drug resistant efflux pump, the MTR. Now Neisseria actually has a couple of different multi-drug resistant efflux pumps. The MTR one's the only one that's clinically important for humans. You can see right here, as of 1 year ago, well, this study was published a year ago. I'll go ahead and tell you about the study. First of all, the multi-drug resistance gene has, in E. coli, because it's the most studied, they created a chemical that could shut that thing down. At least it would do so in a culture. Now, it hasn't really moved beyond that because they haven't been able to explain how the chemical works until a year ago. They finally were able to find out, hey, this is the mechanism by which we were able to shut this thing down. And so now there's more development into shutting down these efflux pumps in order to allow the antibiotics to regain their abilities to kill the cells. Okay, next one, altering antibiotic targets. You remember linezolid will bind to the 23S ribosomal subunit of rRNA. It just so happens, I don't know if all bacteria have this, but it just so happens that strep pneumo and Staph aureus both have several copies of this specific gene. If just one of those rRNAs get mutated, then those two bacteria, those staph and strep, will survive. They will be able to withstand the linezolid antibiotic. That colony will be rapidly selected for, and then it will just take over everything. What I think we're supposed to know, though, is in that situation, whenever you get down to that one colony and you have one gene and there's, maybe 15 or 20 of them that could have mutated, but only one did, you get recombination of all of those genes through homologous recombination, and so eventually that mutation gets spread to multiple genes within that organism. Okay, transformation, you guys remember what transformation is? Transformation is I have these really evil bacteria over here and then I have these really not so evil bacteria over here. I killed this one and he spills his DNA out, and this one soaks it up and becomes evil. And this is how strep pneumo ended up getting some penicillin binding protein mutations, because it got the mutations from strep mitis. And it pulled in the naked DNA from strep mitis after it died, and it caused its own penicillin binding protein to interact and to recombine with that naked DNA. And when it did, it became a mosaic. It got a penicillin binding protein mosaicism. And here's our little guy again, Neisseria gonorrhea. Neisseria gonorrhea got extended spectrum cephalosporin resistance. It's the PIN-A mosaicism. And so a question on your test is going to be, or it might be, I'm guessing, is that, hey, how did Neisseria gonorrhea get a penicillin binding protein mutation? And the answer is going to be it got it through transformation. And here's the problem with that. What's the standard treatment for gonorrhea? It's ceftriaxone, a third-generation cephalosporin. So the review article I was reading, it was published in December 2014 online, published 2015, but it's recent, and it's saying that there are some pan-resistant strains of Neisseria gonorrhea at this point because of this new mutation. So besides C. diff and the New Delhi metalloprotease, this is one of the top microbiology problems. I went ahead and threw a little bit other, MEC-A and MEC-C genes are what give the transformation for MRSA, giving you PBP2A. So we got to ask ourselves really quick before we move on, what are we talking about here? We're talking about altering the target of an antibiotic. So a penicillin binding protein, let's just pretend that this is my penicillin binding protein, and then I have this beta-lactam that goes inside, and it blocks it, and it keeps it from doing its job. And so we're talking about altering this penicillin binding protein. Or we're talking about altering this 23S ribosomal rRNA. Now I have to say, if you were to give a brand new bacteria the erythromycin ribosome methylase, that would be a mutation of some sort. But that mutation doesn't do anything to the antibiotic. It doesn't do anything genetically to the antibiotic's target. What it does is it will cause post-translational modifications to the ribosome, and that will prevent the binding of lincosamides, of macrolides, and of streptogramins. Aminoglycosides still have some activity. Aminoglycosides bind like 800 places, so they still, can work. Okay, last of all, let's talk about inactivating antibiotics. The classic thing is the beta-lactamase. And so here I've drawn my penicillin binding protein, and now I want to get my beta-lactam in there to block it. But wait a second, before my penicillin binding protein gets there, it comes and it gets chopped to pieces, so beta-lactamase. Beta-lactamase stops it before it gets there. You don't have to alter the penicillin binding protein. And so what do? We use beta-lactamase inhibitors. And before long, you get extended spectrum beta-lactamases. So then you start using carbapenems. But then you get beta-lactamase with carbapenemase activity. So then we use more drugs, and then you start getting the New Delhi metalloproteases, and you can't throw any type of attack of a cell wall at these things, they just won't work. On the other hand, you have aminoglycosides over here. These are huge molecules, and they just have hydroxyl groups everywhere, and all you got to do to turn one of these things off is just attach something to its hydroxyl group, and it stops working. Last thing I want to say, now I'm doing this from the cuff of my sleeve, so I may have a mistake here, but I want to go ahead and remind you that you have a couple of other antibiotics. So let's talk about how microbes are able to make nucleic acids. They don't take in folate in their diet; they make their own folate. So these PABA, they have an enzyme called pteridine synthetase, and that converts the PABA into dihydropic acid. I'm just going to put DH acid, dihydropic acid. Then they have dihydrofolate synthetase. Dihydrofolate synthetase converts it into dihydrofolic acid. I should have dihydrofolic acid. I'll do that. Dihydrofolate reductase will then go and convert this into tetrahydrofolate. So this step right here, this can be stopped by something called trimethoprim, or TMP, as it's usually said. And this step right up here at the very top, this is stopped by sulfamethoxazole, or SMX, or other sulfonamides. And the point I'm making here is that this actually can be worked around too, so that you can just find another path to making your tetrahydrofolate. So an alternate metabolic pathway is another option for antibiotic resistance that hasn't been mentioned in some of my other reading so far this week. This is just in case you forgot the mechanism of action of all the antibiotics. And here's 3 really good sources if you want to go out and read something. Hey, sorry, I probably took way longer than it should have. I hope you guys learned something from it. Kick me in the rear end if I need to do something better.